Actually, I don't want to do that. Hey, can people hear me?
Yes, I can hear you.
Good. Thank you, Haley. Let's give people a minute to log in and we'll begin. Oh, it doesn't look like shaped very well today. All right, you should be seeing my screen now. So today is April 30th, and we should be covering Lab 5, Streaking for Isolation and Culture Media. On Thursday, we'll do Lab 6, Dystaffusion Antibiotics, and I'll tell you on Thursday to read the unknown project instructions. I'll show you where that is on Thursday. Any questions about what we're doing? If not, let's go to the lab. Lab 5, cultivation of bacteria, culture media, inoculation, and isolation. As is always, there's a little bit of reading before the lab. Today it's really short. A few pages on culture media, and then a page on obtaining pure cultures. There is no virtual lab today. We've removed it because it required the use of Flash, and Flash is no longer supported. It doesn't work. But there are some video clips in today's lab. Upon successful completion of this lab, students should be able to, 1, understand the importance of proper specimen collection and transport to the lab. Come on. 2, explain the 5 principal steps used by microbiologists to process specimens. 3, differentiate between various types of culture media. 4, explain the purpose of streak plate culture techniques, aka streaking for colony isolation. And 5, define the following terms: selective media, differential media, and reducing media. Any question about any of that? All right. So the ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, drying out, and some to exposure to oxygen. For example, anaerobes cannot usually be exposed to oxygen and therefore require a special transport medium. Once a specimen is received in the lab for microbiological culture, microbiologists employ the 5 principal I steps to process it. First, there's inoculation. Second, there's isolation. Third, there's incubation. Fourth, there's inspection. And fifth, there's identification. In inoculation, you place the specimen on an artificial culture medium to initiate growth, meaning you grow it either on a petri dish or in a test tube. 2, you then isolate it. You have spatial separating the microbial growth into distinct colonies, and usually you'll then streak out each colony to make sure you have a pure culture. 3, there's incubation, placing the culture medium in an environment to facilitate growth. Usually, we incubate our plates or our tubes in an incubator. 4, there's then inspection. Examine the culture for presence of microbial growth, and then you can make some early, get some early characteristics from the species, but you will not be able to identify it until 5, identification, performing specific tests to ID any pathogenic microbe that is isolated. And surprisingly, even in labs today, most labs identify bacteria on performing the specific tests. I thought when I first was teaching microbiology that labs today would do it by genetic analysis, but genetic analysis is not done routinely in microbiological culture. It's still done the older way, where you actually run different tests to identify the species. It's only if that procedure fails that you do like DNA sequencing to identify the species. These 5 steps will be described in detail using different types of culture media. When we're talking about culture media, you should already be familiar with general purpose media. That would be nutrient agar or Na, nutrient agar with yeast extract or NAY, Naye, or tryptic soy agar, TSA. These media allow many organisms to grow, meaning they're not selective, they're cheap, they're readily available. To promote the growth of fastidious microbes, those that have very specific nutritional requirements, you cannot use a general purpose media. You must use an enriched general purpose media. For example, when you add 5% sheep's blood to the growth media, we then call it blood agar, and that would be an enriched general purpose media, allowing many, but not all, fastidious organisms to grow. An example of a fastidious organism would be Streptococcus pyogenes. It will not grow on TSA plates or NA plates. You have to grow that organism on blood agar or a media where you've added specific growth requirements to the media before Streptococcus pyogenes will grow. And the sheep red blood cells add the requirement that Streptococcus pyogenes requires before it will grow. Any question about any of that? All right, so those two different medias. Let's talk about selective media. This is a media that contains chemicals that inhibit or halt the growth of some organisms, but do not inhibit other organisms. Keep in mind, a selective media, when you use it, that first, it may not necessarily be perfectly selective, meaning given enough time, an organism that is selected against may grow and form colonies or a liquid culture, even if the organism is selected against. And second, even those organisms that are supposed to grow on the selective media may grow smaller colonies or may grow a little slower on the selective media than it would on a general purpose media. An example of a selective media is 7% sodium chloride. It has 7% sodium chloride in it. Most organisms will not grow above 2% sodium chloride. So it'll be selective for salt tolerating organisms. Any question about any of that? There's also differential media, and this is media that contains chemicals in it that produce visible differences among different groups of organisms when they're grown in that media. For example, if you had 2 different species that differ in some characteristic, they may look different on the plate of differential media. These differences are unrelated to how well the organism grows on the media. They're usually dissimilar bacteria that visually distinguish from each other, meaning they may have different colors or they may do something different on the media. An example of a differential media is blood agar, which allows you to differentiate between a hemolytic organism and a non-hemolytic organism. A hemolytic organism is one that can lyse red blood cells. Non-hemolytic is an organism that does not lyse red blood cells. Any question about that? All right. Lastly, there is a reducing media. And these media are for promoting the growth of anaerobic bacteria because low or no oxygen conditions are required for these organisms to grow. So you need a reducing media to grow these organisms. You can also call that an anaerobic media. It's important to note that a culture medium can be classified into more than one category. For example, McConkey's agar is both selective and differential. Blood agar is both an enriched general purpose media and a differential media. Any question about any of that? Here we're looking at McConkey's. It is a selective media, meaning that if the bacteria can grow on the plate, it is likely to be gram-negative because McConkey's media selects against gram-positives. So the gram-negatives grow. And then it's differential on the basis of the species' ability to ferment the sugar lactose. If the bacteria can ferment the sugar lactose on McConkey's agar, it will appear purplish or purple pinkish in color from the change in the pH of the media from lactose fermentation, meaning it's slightly acidic. It'll be acidic and it'll turn purple in color. If the bacteria do not ferment the sugar lactose, then there will be their normal color, which here will be either white or off-white. Both bacteria can grow on McConkey's, so both bacteria species are gram-negative. A gram-positive probably would not grow on McConkey's media. Any question about any of that? All right, I've mentioned that blood agar is both an enriched media. You can get Streptococcus pyogenes to grow on blood agar, and it will not grow on a general purpose media. So it is an enriched media. It is also differential on the basis of the hemolytic ability of the bacteria. You can have bacteria which are non-hemolytic. They're called gamma hemolytic or gamma hemolysis. Let me blow this up. And you can see that the red blood stay red all the way up until the bacteria, which are sort of a whitish color here. There are also alpha hemolytic bacteria, and these are bacteria that can lyse some of the red blood cells, but they do not lyse all of the red blood cells. Alpha hemolysis either looks like a darkening of the blood or a greenish tint to the blood. And let me blow that up even further. You can see around the off-white color of the bacteria in the alpha that there is red outside of it, but there's a thin ring of darkening. to the blood, and that's from alpha hemolysis. The darkening is most obvious inside the alpha because all of it's dark compared to here. And that's partial hemolysis of the blood. There's also beta hemolysis, where the bacteria, this might be Streptococcus pyogenes, I'm not sure what the bacteria is here, but it is white in color in the beta, and around it we have clearing of the blood. Around it further we have red blood. Oops, sorry, didn't mean to do that. Hopefully that didn't change anything in the slide. Yeah, it did, it moved that. Oh well, move it down. When you were to hold this up to the light, you can actually see through the clear regions of the hemolysis. This is called beta hemolysis or complete hemolysis. Usually what's seen around beta hemolysis is a ring of alpha hemolysis, meaning you have this darkening of the blood there, and that's partial hemolysis, which is normally seen whenever you have beta hemolysis. Any question about that? All right, this table is giving you the different types of media: tryptic soy agar, nutrient agar, and nutrient agar with yeast extract. That's abbreviated TSA, NA, and NA. They're all general purpose media. Then we have blood agar, which is TSA with 5% sheep red blood cells added. It is a differential and enriched medium, meaning it's an enriched general purpose media. It will grow a finicky or fastidious organism, and it's also differential on the basis of its hemolytic activity. There's chocolate agar, which is abbreviated CAP. Blood agar, by the way, is abbreviated BAP. CAP or chocolate agar is the same as blood agar, except you add the blood when the agar is straight out of the autoclave, and that'll be far too hot for the blood to survive. And so the blood will be lysed by the high temperature, and that will change the color from red to brown, why it's called chocolate agar. Because it looks like chocolate, but in reality it's from blood. It is a enriched general purpose medium. Any question about that? All right. Then we have McConkey's EMB and HE. These are both selective media, and they're all selective for gram-negative cells, and they're differential media. They allow the differentiation between lactose fermenting bacteria and non-lactose fermenting bacteria. So all three of them are selective for gram-negative cells, and all three of them are differential on the basis of lactose fermentation. They will be different colors on different plates if they can ferment the bacteria. And if the bacteria is not fermented, does not ferment the sugar lactose, the bacteria will have its normal color. McConkey's is one I remember. If the bacteria can ferment the sugar lactose, it'll be a purple or pinkish purple color, and it'll be its normal color if it cannot ferment the sugar lactose. Then we have phenyl ethyl alcohol, PEA. This is a selective media. It selects for gram-positive bacteria. It selects against gram-negative bacteria. The alcohol, phenyl ethyl alcohol, gets inside the outer membrane of the gram-negative cell and tends to prevent those cells from growing. Mannitol salt agar is a media which is both selective and differential. It is selective on the basis of salt, meaning it has 7% sodium chloride in it. And if the bacteria can grow in 7% sodium chloride, then it will grow on mannitol salt agar. And if it can't grow on 7% sodium chloride, it'll be selected against. It is also differential on the ability to ferment the sugar mannitol. If the bacteria can ferment the sugar mannitol, the plate and the media around, or the bacteria and the media around it, will turn yellow. And if the bacteria do not ferment the sugar mannitol, the plate will be, the media around the bacteria will be the normal color, which is, it's sort of a reddish orange color. Any question about this table? You'll need this table to work on the lab today. All right, so we had step one. Let's move on to step two, isolation. Louis Pasteur developed the concept of a pure culture. He even developed a method for getting a pure culture, but it was very laborious. And that is, you had to have a liquid culture, and then you made different dilutions of that liquid culture. And then you would take that liquid culture and then plate it in 10 different replicas. So each dilution had 10 different replicas. If ever you had a dilution where only one of the replica had growth and 9 of the tubes had no growth, Louis Pasteur correctly concluded that one or very few cells gave rise to that growth. And so that culture is likely to be a pure culture. And Louis Pasteur was correct. But that was not a very easy way to getting a pure culture. Robert Koch developed an easier way to get a pure culture, and that is you simply streak the bacteria on a solid media called the streak plate method, or the streaking for colony isolation. What you do is you streak out the bacteria on a plate, get up isolated colonies, select one isolated colony, grab the cells from that one isolated colony, and then streak it out again on another plate of media, getting isolated colonies. Once you've got cells that have gone through 2 colony formation events, you have a very high chance that you have a pure culture. It's greater than 95% chance that you'll have a pure culture. And that is good enough for microbiology. So if you want a pure culture, you streak the cells out on plates, not once, but 2 times, so that the cells go through 2 colony formation events. Any question about any of that? And then once that happens, you can say you have a pure culture. And it's greater than 95% accurate, meaning you have a less than 5% chance that you'll have a mixed culture. But there is a slight chance that you'll have a mixed culture. Any question about that? All right, when you're streaking out for colony isolation, what you do is you get a plate and you can use the quadrant or the sector method, where you have a plate and then you streak the bacteria on the first quadrant. Obviously, you sterilize your loop, you cool the loop, pick up the bacteria, streak it on the first quadrant. You'll have the most growth in the first quadrant. And then you streak out the media, you sterilize your loop, cool it, and then you start streaking in quadrant 2. They didn't look like they did it right, but they come here. And then you go through quadrant 2, picking up, or go into quadrant 1, picking up cells on your loop. And then you start streaking up quadrant 2. Okay. This is showing you're only doing, I don't know, 1, 2, 3, 4, 5 different streaks. In reality, you should do about 20 streaks on each quadrant, maybe 20 to 30 streaks in each quadrant. And something that isn't quite shown right here is when you streak out quadrant 1, the highest concentration of the cells will be right here. And as you streak in quadrant 1, you'll have less cells. So your fewest cells will be here. And what you do is to streak out quadrant 2. You start in quadrant 2, go into quadrant 1, but you don't go into this region or that region because this is where the cells are most dense. You want to go in quadrant 1 where the cells are lower in density. And so that would be this streak and that streak and that streak, not this streak and that streak. Any question about that? And then you streak out quadrant 2. And then you sterilize your loop. This one, they're doing it correctly. You start streaking in quadrant 3, and then you come into quadrant 2. They're going too deep into quadrant 2. They should only go to about here and then go back and then come back. And they should only come back about 2 or 3 times. They shouldn't come back. How many times they come into this one? 1, 2, well, maybe only 3 times. That's okay. But don't come in all the way because this will be the highest density of quadrant 2. This will be a lower density. So you come into quadrant 3, come into the low density of quadrant 2, excuse me, and then go and streak out quadrant 3. And quadrant 4, they've done correctly, although they should probably start over here in quadrant 4, move into quadrant 3, where it's at lower density. And then streak out quadrant 4, coming into quadrant 3, 2 or 3 times to pick up cells, and then streak out quadrant 4. The goal is someplace along the line, you'll be putting down isolated cells, which will grow into an isolated colony. It doesn't matter if this happens in quadrant 4, shown here, or quadrant 3, quadrant 2, and if you have a low enough dilution of cells from when you're starting quadrant 1, you could even get isolated colonies in quadrant 1. That did not happen here. Any question about any of that? Now, one cell or multiple cells could have formed this colony. Let me blow this up. You'll note that this colony is much bigger than the colonies around it. And this colony here, as well as that one, is much bigger than the colonies around it. So these colonies are probably formed by more than one cell, in which case they may not be pure cultures. What you want to do is grab from an isolated colony and then streak it on another blank or sterile plate to get isolated colonies. And then once you get an isolated colony coming where the cells have come through 2 colony formation events, you can say that there's a greater than 95% chance that that is a pure culture. All right, then you do incubation, grow the culture under conditions to grow the bacteria. We have incubators, which can be fancy and control, obviously, the time, the temperature, the humidity, and some that are really fancy, like this one here, can control the amount of oxygen as well as the CO2. In our lab, we only have simple incubators, which obviously you can control the time. You put the culture in to grow and then you take it out when it's time. And then we have a trough in here to put water that will provide humidity for the cultures growing in the incubator. That's important because a dry culture, if there's not humidity in the incubator, the dry culture will dry out much sooner than an incubator that is humid. So you may only get 3 days of growth on the plate instead of, I don't know, something like 2 weeks of plate on the growth, on the growth on the plate. Obviously, the incubator can control the temperature. And those are the three things that we can control in our simple incubators. This one here has a bunch of knobs for controlling the amount of oxygen and the amount of CO2 that's available to the cells. For bacteria that are anaerobes, you'd probably want to grow them in a special condition where they receive no oxygen. And that could either be a media that removes oxygen from the media. We'll talk about thioglycolate later, probably later today, if not today, next week or next Thursday. Or an incubator that removes the oxygen and then puts in CO2, such as an anaerobic jar. You then expect the culture for obvious visible signs of growth. Microbiologists can describe the appearance of the colonies by a variety of descriptors, such as the colony size, the colony shape, the colony elevation, the colony margin, that's the outside of the colony. And then you do identification. Once you've obtained relevant microbes that are isolated in peer culture, you run various biochemical tests to try and identify the species. All right, know the terms of this lab. Don't need to know the references. In today's lab, we're not going to be doing any of this actual work. Everything we do will be done online. However, you are held responsible for understanding the material. I sometimes have that. I don't know where that is here, but in today's lab, you're going to be inoculating an agar plate using the streak plate culture method. And we have a little video which you can click here to watch. You only need to watch it up until 4 minutes and 15 seconds. On this plate, note they're using the 3-sector method instead of the quadrant method, which is 4 sectors. And then watch a little video on beta hemolysis, another video on alpha hemolysis, and a third video on alpha hemolysis. This is hemolysis on a blood agar plate. And then we have a little video on McConkey's agar. You only need to watch this video up until 3 minutes and 50 seconds. And then a little video on mannitol salt agar. We no longer have the virtual lab on preparing a streak plate. Let me go to the worksheet now. And then answer the questions. 1, describe natural circumstance where Staphylococcus species of bacteria might benefit from tolerating salt. So give me a natural environment where staphylococcal species can grow by tolerating salt. Obviously, do not tell me something like when you're growing on a plate with 7% sodium chloride, because that's not a natural circumstance. That's done in a lab. 2, explain how hemolysis might benefit a hemolytic organism. So we have a hemolytic bacteria How could its hemolytic ability benefit the bacteria? Three, imagine you have a culture containing a single bacteria species. This culture can be one of many possible species. Describe a scenario where you use mannitol salt agar, blood agar, and EMB plates to help identify the unknown bacterial species. That is, how would you use these plates to help you identify it? For full credit on question 3, you must identify 2 characteristics of each plate that you can use to identify the species. If you only give 1 characteristics for each plate, you'll only get half credit for this one. Any question? Okay, 4. What is the main goal of streaking for colony isolation? 5. What happens to the growth on a plate if you forget to sterilize the loop in between sectors when streaking for isolation? On this question, you must be specific, discussing each sector. You will not get full credit unless you discuss what's going on in each sector of the plate. And note that it says you are not sterilizing the plate between the sectors, meaning when you're going from sector 1 to sector 2, you're not sterilizing the loop. You are sterilizing the loop before you begin sector 1. Any question about any of that? 6. What happens to the growth on the plate if you transfer from stock culture to inoculate each of the sectors? So you pick up from your stock culture and then you streak out sector 1. You then sterilize your loop, pick up from the stock culture to streak out sector 2, etc. Once again, be specific. You must discuss each sector to get full credit for this one. So you need to tell me what's going on in each sector of the plate. 7, suppose you successfully streak a plate and obtain isolated colonies from 2 different species. That is a mixed culture. How can you use this plate to create a new pure culture of only one of the bacteria? Be specific in your answer. 8. In the table below are a variety of agar plate media available to microbiologists in order to cultivate bacteria. Denote whether each medium is non-selective, selective, or differential. Place an X in the appropriate column or columns, meaning some of them can be more than one. I'll give you a hint. We don't have any media that is both non-selective and selective. These are either one or the other because they're mutually exclusive. So some of them can have an X in 2 columns, some of them an X in 1 column. Any questions? And then 9, we have 3 different plates which are inoculated. Which plate was inoculated using the straight plate technique, meaning the streaking for colony isolation? Say plate A, plate B, or plate C. When you're done, finish your answers on the worksheet and only submit the worksheet to the appropriate folder in Canvas. If you submit the lab module, you'll get a penalty, because if everyone were to submit the lab module, my Canvas webpage would soon fill up, and then you guys would no longer be able to submit an assignment. Any questions on anything on this lab? If not, I'll be here until at least 7:45 or until the last student logs out. Once again, if you guys work together, you must have your own answers written in your own words. Okay? Do not simply copy the wording of your study buddy, because that's plagiarism. All right, if there's no questions, I'm going to end it, and I will be here to answer any questions.
I have questions regarding the labs that were the previous ones, like lab 1 and 3, because I know you send it back if like the questions aren't answered correctly. Do you grade those at end of the term, or how do you grade those?
No, I tried to tell you that I was hoping to get to at least lab 0, 0 and lab 01 this weekend, but I
Mm,
ran out of time, so I never got that graded.
okay, and
But I'm
then
hoping to get to it this week, at least lab 00 and lab 01. But I'll do the second grading whenever I have time.
okay, all right, just want to make sure. Thank you.